Today I'd like to run you through an overview of protein synthesis. Anytime you've heard anyone say a gene is being expressed, what they mean is that there's been a protein produced from that. There are two major stages, transcription and translation, and we'll jump into it right away. So the DNA, you already should know, is a long strand of nucleotide pairs. They form a code to make proteins. Now that's not true about every section of DNA. There are some sections that don't code for any proteins. Some of them are regulatory regions that are turning genes on and off. Some of them we have no idea what they do yet. We're still learning lots about DNA, even 70 years after we figured out what the structure was. All right. So again, exciting field to be part of if you want to jump into research. Now, what we used to think, at least, was that one gene or one section of the code makes one specific protein. We've since realized that one gene, one protein isn't true, but we'll talk about that when we get into more details. But for now, you kind of get an idea that at least a section is supposed to make a protein. You can change the gene, and if you do that by causing a mutation, then theoretically you should change the protein product as well. You can go ahead and read about Garrett's hypothesis in your text if you're interested in knowing more about that, but we'll go through some mutations later on in this course in a few days from now. All right, so here's a quick example to take you back to grade 11. Normal hemoglobin carries oxygen very well. It's the right kind of shape. There's about 250 million of these little protein molecules inside every red blood cell, and their job is to just carry oxygen. That's what they do. If you are a sickle cell, then your hemoglobin is badly shaped. And the actual cause of it is, if you look down here, one amino acid has gone wrong in a hemoglobin molecule that is over 100 amino acids long. So one little change in the DNA code was enough to change that very special amino acid, and it was enough to change the function of the hemoglobin. Sickle cell hemoglobin doesn't do what it's supposed to do. It doesn't carry oxygen as well. It doesn't last as long as a normal red blood cell. Normal red blood cells are supposed to last anywhere from 100 to 180 days, and these ones die much quicker than that, which means that you are missing blood or you have anemia. Anemia means that you have less red blood cells than you're supposed to. You can also have changes to the shape of the red blood cells. Instead of being a nice, you can see this picture down here, instead of being a nice floppy disc shape, it becomes more of this yucky sickle shape. And because of that, you're going to get it stuck in blood vessels. And that can actually lead to a lot of pain, can damage body organs. Again, it can lead to anemia because you're not carrying oxygen correctly and you don't have as many blood cells as you should. This can also, if you are not in some kind of a fancy country that has fancy healthcare, anemia can kill you. If you are somewhere deep in the heart of Africa where you don't have that right kind of care, you're probably gonna die young from sickle cell anemia. Why say Africa? Because that's where it's most prevalent. There seems to be some kind of a heterozygous advantage for people who live in Africa because these horrible sickle cells are really bad at carrying the little protist that causes malaria called Plasmodium. And because the little malaria disease doesn't like to live inside these cells, people that have somewhat sickle cell, people that are the hybrids, so if they have some good cell information and then some sickle cell information, they may not suffer so much from sickle cell anemia, because they have some cells that actually do carry proper hemoglobin and can carry oxygen, but their cells are slightly weirdly shaped and the Plasmodium does not like it, doesn't live there. So they have a slight advantage in both cases. They could survive anemia and they won't die from malaria. So you tend to have a lot more people of African descent, especially middle Africa, where malaria is very prevalent. There seems to be a lot more hybrids, and it seems to be linked to their ability to survive malaria. Now, malaria doesn't always kill you, but it can cause complications. All right, so one little mistake is the point. One little mistake breaks that particular gene so that it doesn't produce the right kind of protein, and it clumps together and makes these horrible sickle-celled shapes, okay? Now, just a reminder as we jump back into DNA, that the DNA code could have thousands of nucleotides in that one section. If you've got something like hemoglobin where there's 100 amino acids long, what you're going to learn shortly is that every amino acid has a 3 nucleotide code attached to it. So if your actual product is about 100 amino acids long, then your actual DNA has to be a minimum of 300 amino acids long. And you'll actually see that it's actually probably a little longer than that because certain parts are going to get chopped up, but we'll talk about that when we talk about that. So just a reminder as well, you're going to read DNA from 5' to the 3' direction. This is the sense strand, which makes this the antisense strand. And this is also the coding strand, which makes this the non-coding strand. And this is also the strand that we're never going to use to actually make a protein. This opposite strand is the one that's going to be used as the template to make the mRNA. This one, though, the reason it's called sense or coding, not just because it's 5' to 3', but because it's going to match the mRNA, and you're going to see that shortly. So when we are actually get down to it and we're actually using the DNA, we actually use this template strand when we're writing things out. So for pretty much all of your questions, unless I'm asking you to go from DNA to DNA, I'm probably giving you the template strand, the 3' to 5' version, so that you can directly change that into mRNA, which you'll see that as we go along. So just a reminder, this is when we're starting to have these terms actually be used correctly. It may not have made sense in the last lesson, but they will make sense hopefully in this one. So sense and coding strand, antisense non-coding, also called the template, also called the complementary, also called the anti-parallel strand. I know, isn't it great to have so many names? All right, so jumping into the practice here. Sometimes I might be asking you to create a complementary DNA strand. Well, you should know then that A pairs with T, G pairs with C, C pairs with G, T pairs with A, and so on. If you're asked for the DNA, make sure you use DNA nucleotides. And you'll know that this is DNA because I'm using a thymine. If it was RNA, I'd be using U for uracil. Okay? So again, this would be the coding, and then this would be the non-coding or the template strand. And I know that because the coding is always 5' to 3', and the non-coding is always 3' to 5'. When you're writing these out, remember to write the primes. It's very important for the DNA and the RNA strands that you tell us what direction they're going in so we know which strand you're talking about. Okay, let's keep going here. So this could be your own little simple practice. Feel free to do that on your own time. So the code is stored in the DNA inside the nucleus. Okay? But the proteins that are actually made by the ribosomes, well, those are outside the nucleus. So if you've got your DNA inside here, but you've got your protein machinery out here, well, you're going to need to get this code from here to the outside. The thing is you do not want to take this DNA out of here. The reason it's wrapped in the nuclear membrane is to protect it and to keep it from being digested by things that live out here, the little enzymes that are free-floating or the lysosomes that are out here. You really don't want them getting into contact with your DNA. You don't want to damage your DNA library, because when you damage it, then everything that is produced from it will then be wrong. You'll have all these terribly badly made proteins if you damage your DNA. Okay? It's also a giant molecule, huge, massive molecules. 46 of them are in here, and you don't need all 46 to go out. You just need one section of one chromosome to be copied, and then the copy can go out. It's kind of like, I always compare it to you leaving the books in the library. Leave the recipe in the library and just take the one photocopy of the one page that you need to make the thing that you're going to make. You need to make a cake, just take that one page. You don't need the whole book. And you take the one page, the copy, and you take that out to the kitchen, and then you can cook up whatever you need to cook up. All right? So that's the kind of the idea. So DNA can't leave, ribosomes can't enter, so you need an intermediate or separate molecule to transfer that code from the DNA to the ribosomes. And you've probably already guessed that that transfer molecule, that separate molecule, is going to be RNA. Now, we call it messenger RNA or mRNA because it's taking the message out to the ribosomes, okay? When you rewrite the DNA as RNA language, we call that transcription. So DNA gets transcribed to mRNA. You rewrite it in a new language. That's why we use the term transcribe. Then the mRNA moves out of the nucleus and goes to the ribosomes out in the cytoplasm. Keep in mind these ribosomes could also be attached to the endoplasmic reticulum, which will make it rough endoplasmic reticulum. But either way, it goes to a ribosome. We'll talk more in detail about how the ribosomes actually work in the next lesson. Then the mRNA gets translated. What do we mean? We mean that whatever this code is written here on the mRNA gets read by the ribosome, and then this 3 little letter code called a codon. Every 3 letters is going to code for a very specific amino acid. So that's why the order of the DNA matters. If this isn't right, if the DNA is wrong, then the mRNA will be wrong, and then the amino acid that gets inserted will be wrong. And that's exactly what happened in our first example at the beginning with the sickle cell anemia. One amino acid was wrong because one codon was wrong because one portion of the DNA was in error, and that created the wrong message down here. And then the protein didn't fold properly, etc., etc. Big problems with carrying oxygen. Okay, so you want to make sure DNA is good so that the mRNA is perfect so that the protein code will be done correctly. So that's it. Those are the 2 major stages for protein synthesis. You transcribe, you move it, that's called translocation, and then you translate. And again, transcription is to rewrite, translation is to read it and make sense of that new code. When we are creating the mRNA strand, you're going to see that it is also complementary. So the G pairs with C, C pairs with G, A pairs with U, T pairs with A. But notice there's no T's down here in the RNA strand. Again, thymine doesn't exist in RNA. You have to replace it with the U. So you have the A pairing with a U and the T pairing with the A when you're doing your translating and transcribing, pardon me, when you're doing your transcribing. Okay. So let's keep going here. During transcription, you're going to copy that code from DNA to mRNA. We already said that. I'm going to jump now to a little animation just to show you kind of how things kind of work. So let's start off by looking at a pretty complicated protein. These little round balls that you see there all represent different amino acids. And let's jump right into how this insulin molecule was formed. Just another reminder of structure. You've got your cell, and then you've got your nucleus, and then you got your chromosomes on the inside of that nucleus, and then of course the chromosome is made up of this DNA structure here. Now when we look at the actual section of the DNA itself, when we zoom in, only some portions are considered genes, and these are called the coding regions, and again because they code for a protein. So not all of the regions in an entire chromosome will actually be directly useful for making a protein. Again, we still don't know what some sections of the DNA actually does. In order to get a gene to actually work, we need to actually have these little activator proteins that are going to attach to switch that particular gene on. And we'll talk more about that when we get to talk about operons in a couple days from now. But for now, just know you have to actually have an indicator, something that says you must start the actual process. And this is always going to be called upstream of the gene. So to the left of the gene, and always to the left because you're always going 5' to 3' direction. Okay, so you always start at the 5' end when you're activating so that the actual strand can be run in the 5' to 3' direction. You'll get other transcription factors that will also be included, but again, we'll talk about this in detail. You're going to have the same idea where you're going to want the DNA helicase to unwind the double helix. You're going to want RNA polymerase in this case. It's going to come along. And this here, the big lumpy guy in the middle, is your RNA polymerase. And what it's doing is it's reading the DNA. Notice it's only using the one strand. And what you have coming out the back of it was the mRNA. And if we zoom in on what's actually happening, the DNA gets split by the helicase, and then as it's running through, the RNA polymerase is just adding in RNA nucleotides that match the DNA strand, and the mRNA strand is lengthening out the back of the RNA polymerase. But notice the very bottom strand that you see there is the coding or sense DNA, although it's not being used, which is complementary to the template strand of DNA, which is the middle strand, which is being used. And then notice that then the mRNA also makes sense again, because look how it matches the DNA that's at the very, very bottom. Give you a second to take a look at that. Now, before we actually end up taking this messenger RNA out of the nucleus, we have to do a little bit of work on it first, but I'll talk more about that in the next lesson when we talk more about details in transcription. The third type of RNA that we care about is the transfer RNA, or the tRNA. And notice its shape here as you're looking at it. It's quite literally in the shape of a T. But what you actually have here is one long strand of RNA that's folded in on itself. And if you were to look at the loops here that are formed, the 3 loops, you can see that in between those little sections you have complementary bits of RNA that fold in on themselves. But on the very right-hand side, you have something called the anticodon. And they've blown it up here to make it much larger, but notice it has 3 nucleotides on there. Those 3 nucleotides are anti or opposite to the mRNA. And at the far end, on the left-hand side of this long transfer RNA folded strand, you have an amino acid attached. What we're looking at right here is the final version of the mRNA that's gone out to the ribosomes. And notice we've broken things down into 3s. Each one of those is called a codon. And each one of those codons is going to match a tRNA. And as the tRNA comes in and finds its counterpart, notice that they are complementary. C pairs with G, U pairs with A, and so on. They bring in, one after another, they bring in the amino acids that are attached to them, and then they break off and leave the amino acids behind. And then those amino acids are then able to fold simply because of their different R chains. They'll either fold in alpha helix coils or into beta pleated sheets, and then you'll get your final tertiary subunits, and then those subunits will be added together to make your final protein product. And that is protein synthesis in a nutshell. So now that you've got the basic visual down, let's run through your note again and we'll finish and tidy some things up. Now, just a reminder, you must remember that when you're building RNA, you have to use U for uracil, not T for thymine, because the T only belongs in DNA. It's really, really key that you remember that. So the letters that you're going to use for any RNA code are AUCG instead of ATCG. A pairs with U, C pairs with G. So let's get on and we'll do a little bit of practice down here. Now it is important that you do practice this for yourself, but for now, let me just run through it with you. First thing, you'll have questions like this on your test. You'll say create a complementary, so you know it's going to be opposite to. You're creating the mRNA strand using the DNA template sequence. Notice that it's the DNA here, it's definitely DNA because I'm using T's, is running from 3' to 5'. So this tells me that this is the template strand, okay? Remember that the template can also be called the antisense or the non-coding because it's going to be opposite to what the mRNA code is. And remember that the mRNA code will be identical to the sense strand of DNA, which would have been the 5' to 3'. We just didn't bother to draw that. But if you look down here at my little picture, you can see I've tried to write it out for you. So here would have been the sense strand up above. This is the same as the template down here that we've got, the 3' to 5' DNA. And then the mRNA transcript is going to come in between, and it's also going to be 5' to 3'. And if you look at the letters here, you'll see that it matches the sense strand, even though the sense strand itself is not used to make the protein, or I should say the mRNA code. It is used to try to match and to do any sort of, if you're trying to double check anything, or you would check it up against this strand here. Okay, so the mRNA will be complementary to the DNA template strand, and it will be matching to the sense strand because they both make sense. But notice A and then T and U are substituted. Then you got 2 G's, doesn't look like much like a G, but it is a G. U and T are opposite, and then C is matched. So there you go. That's what we've been meaning by sense and non-sense strands, okay, or anti-sense strands. So here I've just given you 2 different examples. Please try this for yourself and write them out for yourself. Make sure that you're feeling confident about being able to do this. It's pretty easy, but you gotta use some brain power, and you gotta double check and make sure you haven't made any silly mistakes. The main thing would be to remember that A's are always going to pair with the T's, but when you have a U, it'll pair with an A here. So in my DNA language, all my T's become A's and all my A's become U's rather than T's down because I'm using RNA language down here. Don't forget to write the primes. The primes matter because they tell us the direction and they tell us that this is a sense strand, mRNA sense strand. Please, please, please remember to do that. So that's it. That's transcription. Super easy. You're going to be doing lots of this over the next few days. Okay. Then we can go through some transportation. That's when you just get the mRNA molecule to be carried from the nucleus out to the cytoplasm where it meets up with a ribosome. And then you get to the translation section, which I already showed you in the little animation. And here you have the mRNA gets read by the transfer RNA. This is what the ribosome's job is to do, is to grab the right correct tRNA and line it up and let it attach to momentarily, just with brief hydrogen bonding, and it pairs up, and then this amino acid gets left behind. And when we do the translation details in the next lesson, I'll show you more what the actual ribosome is doing. But that's just the basic idea. So you have these codons, which are 3 base sequences in the mRNA, and each codon codes for a very specific amino acid, because each tRNA, so the 3-letter code that's here, you can't see this, but this would have been a little U. This UAC always codes for this particular amino acid. And I'm going to show you a codon chart shortly so you can see what I mean by that. So tRNAs have an anticodon because the anti is opposite to the codon on the mRNA. Okay? Always lines up and matches the mRNA chain. And again, you'll have these guys coming in along the sequence here, running in that same direction the entire time so that they leave the right amino acids behind. And then these amino acids are joined with their little peptide linkages here done by the ribosome, and then you've got yourself a nice functioning peptide chain. The peptide chain will then fold, and then so on and so forth, and you've got yourself your final functioning protein. Okay, here's some more practice I'd like you to do, but I'm going to run through it just very quickly here for the sake of time. But please actually do this on your own. Don't just look at my answers here. Go ahead and try it on your own. It will be beneficial for you, and you can see if your work matches mine. So I'm going to start off always by giving you the template DNA strand, although in your one worksheet you will be given first the coding strand, then asked to create the template strand, then jump through these hoops. Okay, so if you see 3' to 5', you know it's the template, you know you're jumping straight to the mRNA. So you do your opposites. T pairs with A with U, C with G, A with U, so on and so forth as you're going through. Don't forget to write the primes, 5' to 3'. It matters, okay? But this time what I'd like you to do is then consider which tRNA anticodons would have to slide in. So keep in mind there's a big T-shaped structure sitting here with this amino acid attached to the other end. MET stands for methionine. We use 3-letter codes to show the names of the amino acids so we don't have to write them out in full. But just, you'll get to that in a second. So we're going to take the DNA, we're going to do the mRNA sense strand, then we're going to show the code for the tRNA antisense and which amino acid would have been attached to that. Now, notice here, I've made a little note, you do not need primes for tRNA. You only need the primes for the DNA strand, 3' to 5', and then showing the 5' to 3', because these are long chains. These are not long chains for the tRNAs. I'll show you again. They're chains folded in on themselves, but the only part that matters is this little section right here, these 3 little anticodon nucleotides here are the only ones that matter. So we're not really caring about whether it's 3' to 5' or 5' to 3'. It would be lined up probably 3' to 5' on the end of that guy, but we don't care. It doesn't matter because it's a momentary opposite complementary relationship. Then this guy will fall away and leave this amino acid behind. So it really doesn't matter. Now the codon chart that I'm going to show you, how we found out that this was methionine and phenylalanine and so on, is we actually use something called the codon chart, but it's an mRNA codon chart. So these tRNAs, this would be a waste of our time for us to always write these out. I'm only going to ask you to do that for a couple of questions in your homework, but you're always going to jump from the mRNA strand straight to the amino acid chain. But let's jump to the codon chart and I'll show you how to use it. So this is the mRNA codon chart. All right, looks a bit crazy at first glance, but it's really not. What you're going to do is just look for the 3-little letter code that you're wanting to look for, and then you're going to jump from there. So our actual first little code was actually AUG. So there's A, second letter U, third letter G. I can see AUG written right here. So I know that the AUG, it's going to start off with methionine, MET. And you're actually going to find that every single mRNA chain always starts with methionine, and I'll tell you why in the next lesson. But you need to know that. You're always actually going to have AUG as your starting mRNA code. So let's continue translating then the final thing here. So we see that we have a UUC. Again, we're using the mRNA. UUC, I go over here and I look for where is it? UUC. Oh, phenylalanine. Okay, PHE, done. GAA, go over here. Where's GAA? GAA. Oh, it's glutamic acid. GLU. Great, write it down. Then you're going to see CGG. Let's go find that. CGG is an arginine. So ARG, done. And then UAA. Well, now when you go up and you see UAA, you actually see stop. Stop is not the name of an amino acid. Stop means that there is no amino acid that will actually be found on any tRNA. There's no tRNA that exists that has UAA on it. There's also none that has UAG and none that has UGA. And again, I'll show you how that kind of works out. But the basic idea is that if there is no amino acid coming into this location, no tRNA to go there, then there's nothing to place here. So even if this code on the mRNA continued, when you brought in the next tRNA, this new amino acid wouldn't join the last chain. This last chain would pop out the other side of the ribosome and it would be a separate chain. So the stop literally means that is the termination or the end of that amino acid chain. Okay, so it's useful then to know if the stop codes are UAA, UAG, and UGA, and if the start code is AUG, the methionine down here, then it's also useful to know what the actual DNA sequences would be so that you can start to recognize. So basically, for example, you would see AUG as your start codon, which means TAC on the template strand will always be the start on the DNA. And if UAA happens to be a stop codon in the mRNA, doesn't code for anything, then ATT is the DNA version of the stop codon. And it can save you a lot of time if you memorize TAC. You probably will anyway because you're going to be looking for this over and over again. But committing ATT to memory is also a little bit useful. But because there's 3 of these codes, let's quickly jump and see what the other codes would be if we translated them into the DNA language. So once again, here's our start sequence. AUG always codes for methionine, and its opposite in the DNA language would be TAC. UAA, UAG, and UGA are all versions of stop codons, and then these would be their DNA complementary counterparts. So it's worth knowing them. You'll always be able to work them off of the mRNA chart anyway, but especially TAC, really useful to know. So if you had a DNA sequence and you needed to translate it, you would transcribe it into mRNA first, and then you would translate it into the amino acid chain. Then you would have to be looking at it. Notice it's 3' to 5', so that tells me it's template. I would be reading along and reading along until I saw the TAC. Then I would know to start my mRNA code. This is exactly what the RNA polymerase will do. It'll read. As soon as it sees the start codon in the DNA, it begins to add in the little RNA nucleotides to make this, to transcribe this mRNA chain. So please make sure you start at the start. When you're doing your mRNA, you start at the start, okay? Then you can continue on for the rest of the chain, no problem. Then, when it's time to go through and actually translate it so that you're going to turn it into amino acid, you would make sure that every 3 is broken up, however you like to do it. Some people like to draw lines, some people like to do dots, whatever works for you to show that they are in groups of 3 so that you don't mess up the sequence. So you would do the same thing I just showed you, where you read the AUG and you're going to jump straight to whatever amino acid is supposed to go there. AUG always codes for MET, so that one's easy. Notice I haven't bothered to put in the tRNA code, the anticodons down along here. You're going to find very shortly that gets very tedious. That's why I'm only asking you to do 2 of those questions in your homework. And you're going to want to just make sure that you jump straight from the mRNA straight to the amino acid, save yourself some time. So notice we've got a code for amino, code for amino, code for amino acid. But then here's our UAA. That's a stop codon. Nothing goes in here. There is no amino acid for this codon, so the chain's going to terminate. So please remember, you start at the start. Okay, start at the start when you're trying to make your mRNA, and then stop at the stop. Don't try to keep adding amino acids in after you've had a stop. Start at the start, stop at the stop. Okay? Please make sure that you go through and you answer this question right here. This is a very good thought question to get you to understand what's going on. But I also want to show you something pretty neat about the mRNA codon chart before we stop this lesson. So here we are staring one last time at our beautiful mRNA codon chart. And I want you to notice the position of the bases and look how often we actually care about what position they're in. If you have the first as a U and the second as a U, you're either going to be phenylalanine or leucine. But notice that if you have the first as a U, the second as a C, and any of them, U, C, A, or G in the third position, notice it still codes for serine. So it doesn't really matter what the third codon is in this particular case. It's always going to code for serine. Same thing here. Look, CCU or CCC, CCA, CCG. The third codon here doesn't really matter. It's going to code for proline either way. That's the same for leucine, valine, alanine, glycine, arginine, all these guys. There are a few where it will make a difference, and those are the ones that share the box. So, a U versus a C, you'll probably end up with a phenylalanine, but if it was an A or a G at the end, it could be a leucine. And notice there's actually 6 different codes that code for the leucine itself. Okay? There's only one start codon. There are the 3 stop codons that don't code for anything, and tryptophan also only has 1 code for tryptophan. But all these other amino acids have 2 or 4 or sometimes even 6 different codes that'll still code for it. Why am I pointing this out? Because it matters what position you have the mistake in the DNA. If you had the mistake in the third codon of the DNA, and you could have gotten any of these mRNAs as a result, it would not have changed the amino acid code 1 iota. It would have been identical. You would have still gotten serine. So you could have a slightly different DNA code as long as it's the third codon, and you might not even change the actual product itself. But if you were to change the first letter, so a U being changed to a C or an A or a G, you can see in all of these cases, you would have a very different result. So it matters which part of the codon actually got damaged in the DNA. You may or may not actually see a mutation happen as a final product. The mutation will be in the DNA, but it won't change the actual final product for the protein. Alrighty, so just keep that in mind. We're going to be studying some mutations going forward, but I just wanted to highlight that while the chart is brand new and fresh in your mind.